actually, because the first thing to say is to welcome everybody. I think when we look back at over the last little bit of time, actually these node showcases were sort of one of the things that we did that was really valuable. And I think people really found it very interesting to find out what other people are doing. And I know lots of, I speak for myself, connections, have come out from these of people's, areas of work they weren't aware of. So we thought we'd run it again for that precise reason, because it's just really interesting. So at that point, let's move on to the actual node showcase elements of all of this. And I don't think Anna and Nikita have had any formula for who comes in what order. I think it's random. So don't read anything into this. But the first node is the Express node.
Thanks, Dave. Can everyone hear me? And I think I can drive the slides for everybody from across Teams. Is that working? Slides are advancing. Yeah, I assume they are. So yes, it's a pleasure to talk about progress with the Express node. So we kind of seem to have got to the 50% mark through the study quite quickly, but I think we have made progress and look forward to really taking things forward over the next couple of years. So study team, so this is the sort of core study team, if you like, of the Express group. So myself, James Rowe and Tim Rittman in Cambridge. We're working on imaging, so dedicated research imaging, but also sort of harvesting routine NHS scans to enable early diagnosis of these conditions. Michelle Hugh in Oxford, working particularly on digital biomarkers, so deployment of smartphone devices that may help with early diagnosis of these conditions. Yoav Ben Shlomo, epidemiologist in Bristol. Riona Fumi and Isabella Carrascos in UCL, core study team. Ed Jabari working on biomarkers. And I would say a very important part of the study is the integration and support that we get from charities, particularly the PSP Association, MSA Trust, and we're having frequent meetings with them and updates on the progress with this study. So the Parkinson's plus conditions, these are a sort of more malignant, more rapidly progressive, much rarer subset of conditions that are frequently misdiagnosed as Parkinson's disease. So this is, from our perspective, very kind of under studied patient group. So particular challenges are underdiagnosis. So some patients may never get diagnosed having these conditions. Lack of awareness, provision of support. So these conditions are much less well supported than other neurological conditions like motor neuron disease. Understanding of disease biology and early disease stages. So most studies have been undertaken in people with well-established disease. And in fact, the average delay from symptom onset to diagnosis, and these are progressive neurodegenerative conditions, around 3 to 3.5 years for these Parkinson's plus conditions. And in fact, the life expectancy is about 6 to 7 years on average for these conditions. So that means that for 50% of disease course, people are undiagnosed, they don't have the right diagnosis, they're not available for not able to enter clinical trials or clinical research studies. In the second half, of course, unfortunately, people are deteriorating quite rapidly with deteriorating speech, swallowing, and mobility. So that's a real barrier for translational work with patients. So the Express study follows on from our earlier study called the Prospect study, which was a cross-sectional and longitudinal study of patients with established disease, but here trying to focus on early diagnosis. So the main goals of our study was to establish and to maintain a clinical research network of sites in the UK that can participate in these studies, to have a platform for early diagnosis for biomarker and therapeutic trials, to enable some discovery science, some basic science that will be built on top of this node, to establish collaborative projects and trials with basic science investigators and with industry. So the current situation is that we have 19 active Express sites in the UK. So this is the distribution of sites, and this includes some sites which are very experienced with doing in-depth research in these neurological conditions, some sites which are less experienced, and actually with some of our sites, these are led by non-medics, by research nurses or research nurse specialists acting as principal investigators of these sites, which I think is great. To try to enable, and often they have a very good knowledge of the patients going through their centre. So there's a mixture, if you like, of different types of academic and more sort of real world centres, if you like, within our network. So the Prospect Express study, the Express arm, the aim is that people are assessed within their first 12 months of having been assessed in secondary care for Parkinsonism. So I think certainly looking at UK Biobank and looking more broadly at general practice databases, it would potentially be possible to recruit people using diagnostic algorithms in primary care. Because of the rarity of these conditions, the sort of that's quite a difficult and challenging thing to do. So for us, what we really focus on is people when they first enter the secondary care system and trying to pick up the markers which indicate they're going to have one of these more malignant Parkinson's plus conditions. That's what we're really trying to do within this study, and to enable early diagnosis. So the idea, of course, with the study is that there's consent for patients. There is some clinical assessment. We tried to make this as far as possible something that can be done as a minimal extra assessment within a routine clinic visit, collection of blood for DNA and other biomarkers, and then the MRI imaging part, James and Tim leading in Cambridge, and the digital part, Michelle leading in Oxford. And of course, for movement disorders broadly, Parkinson's disease, ataxia, Parkinson's plus conditions, there's of course a huge interest in digital biomarkers that actually can measure people's mobility, their falls, how that changes the disease course, both for early diagnosis of these conditions and for progression, what happens over time, gathering sort of real world data outside the clinic that can enable early diagnosis. So we've had some iterations and piloting of the protocol, and currently we're up to 46 people who've been recruited through the platform. So 30 through the Express core, so that's light touch recruitment, and 16 through the deep biomarker phenotyping. Our actual goal in the original proposal was to recruit about 100 people to the Express biomarkers arm of this. But clearly, once the platform is established and running efficiently, we would like to, and are applying for more funding to try to expand that part of it, because this is a bit undernumbered at the moment. It will need certainly bigger numbers for this. One of the things we do as well within this study is to invite people to register for postmortem brain donation. That is the gold standard for diagnosing these conditions, and that of course enables us to compare diagnostic biomarkers with patients who unfortunately died from these conditions and their postmortem findings. And we have a fantastic brain bank network in the UK which has been involved particularly in Parkinson's disease, but also studying these Parkinson's plus conditions over the years. So trying to link up the biomarkers collected in life with the autopsy findings in these patients. So in terms of we have active academics doing lots of different research. So people are putting in additional grant proposals based on what we're trying to do in the Express network. And this highlights some of the add-on funding bits that have been successful. So Tim Rittman has been successful getting more funding for imaging from Alzheimer's Research UK. James Rowe in Cambridge has philanthropic donations and supported by the BRC, and a philanthropic donation to the Cambridge Centre for Parkinson's Plus. Ed Jabari here at UCL has an MRC Clinician Scientist Fellowship, which has just started to look at biomarkers across the range of Parkinson's plus disorders, but particularly hopefully using samples collected within the Express study. We have funding from the Global Parkinson's Genetics Programme for genetics. Michelle Hugh has funding from PSP Association Digital Biomarkers. We have some other supplementary funding applications that are in preparation or progress to try and add on to the sort of incredible infrastructure really that's been funded by this MRC NIHR platform that enables us to really try to add on extra value to the network we're establishing here. So these are some of the sort of discovery science things that are working. Some of this, of course, predates, or the sample collection predates Express, but this is some of the things happening within the network. So just going through a lot of interest in these seed amplification assays. So that's the ability to look at seed-competent abnormal neurogenesis proteins, particularly in spinal fluid. So that's particularly the work Ed Jabari's leading, looking at the moment at synuclein, but hopefully moving on to 4-repeat tau protein and TDP-43. Some of these high-throughput proteomic platforms are being studied within this patient group. This paper from James Rowe Group rates the new LISA platform, looking at proteomics in patients with these conditions. different MRI imaging patterns, some of the clinical data that's been studied by Michel and Sanjay in Oxford, and some sort of genetics looking at the overlap between Mendelian genes that cause similar syndromes and the patients that we're recruiting through these networks. Riona's been particularly looking at re-diagnosis. So what happens when someone comes to clinic, first get diagnosed as Parkinson's disease, then get re-diagnosed to have another condition, and looking at the kind of looking back to some of our studies of the sort of baseline determinants that indicate that someone's got one of these rare neurodegenerative conditions. And obviously that may be helpful as we sort of deploy new ways of diagnosing this going forward. So this is from two big Parkinson's studies in the UK, the ProBAN study in Oxford Parkinson's Discovery Cohort, looking at patients who were recruited in all good faith by neurologists at the start of the study with diagnosed Parkinson's disease, then turned out not to have Parkinson's disease, not because they weren't very good at diagnosing, because other features emerged which indicated that they had one of these malignant Parkinson's plus conditions. That's the sort of re-diagnosis project. This is actually looking at a very well-known biomarker in neurology, neurodegeneration, neurofilament light, together with clinical features, and looking at how that might be predictive of having an alternative diagnosis at baseline analysis. So this is work that Ed's been leading, looking at seed amplification assay. This is actually looking at 4-repeat tau. This is from the brain of patients with PSP, and looking at an in vitro assay of abnormal seed-competent 4-repeat tau protein. So what we're seeing here on these curves is thioflavin T fluorescence that indicates there's abnormal aggregations happening, and these are serial dilutions of brain being inoculated and the time to the initiation of the abnormal aggregation pattern. And you can see that in these brain samples from patients with PSP, you get a very robust pattern of abnormal signal as compared to patients with Alzheimer's disease and patients with another tauopathy, Pick's disease. So this looks very, very good in brain tissue. The difficulty is translating this into a CSF assay that can be used in living patients. So that is proving very challenging, and is something that Ed is trying to crack in terms of developing a CSF biomarker of these conditions in collaboration with Byron Corhy at the Rocky Mountain Laboratories. It's a Prion laboratory, an NIH Prion laboratory in the US. We're still interested in families, so we do see families with multiple people affected with these conditions, and obviously this is a real kind of point of overlap with some of the other nodes in the rare disease research network, which are much more Mendelian. So we are seeing families with these conditions. Some of these families have mutations in the tau gene, but some of them don't, and we think there probably are more genes to discover in this patient group, more Mendelian genes to discover that have not been discovered yet. So this is obviously difficult in people with very late onset neurodegenerative disease, where it's difficult to recruit large families, but we're continuing to follow up these families through the network. And I'm still optimistic that we will find some new genes within this patient group that will give real sort of insights into the underlying biology of these diseases. So this is some of the imaging type work. This is actually work that's done by Will Scotton at UCL, looking at the progression of atrophy in patients with these conditions. So what happens with different atrophy patterns over time. This is stuff that's been done through observational studies, but obviously this is very relevant for future clinical trial design, where you can look at the progressive loss of brain tissue, of course, through atrophy that happens as these conditions progress over time. So we think that this sort of approach will be helpful for future clinical trials. We're very interested in different patterns in non-European individuals. This is data from brain banks, where we've actually used principal components analysis to define people's ancestry genetically. This is looking at Lewy body disorders with synuclein and PSP with tau. This is looking at genetically defined Ashkenazi Jewish patients, non-Ashkenazi Jewish, non-Finnish European patients, and South Asian patients, and actually showing that within, albeit very small numbers, in terms of people who've been recruited to brain banks, that South Asian patients have a much higher proportion of PSP, of tauopathy, and we collaborate with colleagues in India and other parts of the world who see a lot of these patients. We see a lot of South Asian patients in London with PSP who are engaging with research studies, And I think that this is an evolving story, that there may be different types of neurodegenerative processes that are happening in different ancestry groups, which is clearly very important. So networking, we have close ties with the PSP Association, MSA Trust, frequent talks, meetings with patient groups, and the PSP Association have supported a lot of our research over the years, and we really that tie with the patient group is very important in terms of publicizing research and also encouraging patients to be involved with research studies. So industry collaborations, there's an ongoing antisense oligonucleotide study for synuclein, MSA. There's an ongoing study of a drug which alters post-translational modification of tau called the PROSPER study, which is fully recruited. There are likely to be further antisense oligonucleotide studies for tau that are coming up in patients with PSP. I think this is another point of overlap, of course, with the with several nodes, including the UpNat node within the Rare Disease Research Network, and this is kind of a point of contact between a slightly more common rare disease, but patients are progressing rapidly, and the kind of invasive nucleic acid therapy is being kind of proposed as a disease-modifying treatment for patients. So this is a point of contact, if you like, between the conditions we're interested in and some of the much rarer single gene disorders that are being studied across the network. So in terms of what we're doing, focusing on rare late onset disease, focusing on the diagnostic process, which is largely non-genetic, and trying to improve recruitment through non-specialist centres and our links with disease charities. And the challenges, I think one of the particular challenges overall is the rapid progression of these conditions, and also delay in NHS recruitment pathways for patients. So that is a major that's a major barrier, I think, to recruiting patients for these into this study. Yeah, so that's my presentation. Thanks.
Okay, brilliant. Well, thanks a lot.
Thanks.
Absolutely fascinating. So let's move on to, if that's okay, to the ELSI node.
Hi, can everybody hear me?
Yes.
Okay.
And I'm not being rude by not introducing the speakers. It's just that Nikita and Anna haven't told me.
No problem at all. I will tell you some more about the ELSI node then. So my name is Faye Johnson. I'm a research associate on the ELSI node in Manchester, and I joined the team in January of this year. So ELSI stands for Ethical, Legal and Social Issues in Rare Conditions Research and Clinical Practice. And just an overview of our node then. So we are a partnership between researchers and projects in Manchester, Oxford and Cardiff. And we work with patients, families, communities, clinicians, academics and industry to explore those ethical, legal and social issues related to advances in genetic technologies. So our aims are to improve the design of clinical trials for rare conditions, to understand the psychosocial impact of living with rare conditions, to highlight barriers to mainstream access to genomic testing and research and develop solutions, and to develop proposals for good clinical practice. So as I said, we are a partnership of researchers across 3 different areas and 3 different projects. So the project that we're working on in Manchester is all about impact and outcomes in rare conditions clinical trials. And our ultimate goal is to develop capacity for a meaningful set of core outcome sets and patient-reported outcome measures that will capture the needs of patients and family members. Project 2 with the Oxford team is to understand the experiences of those living without a diagnosis and consider ways in which people and families could be better supported. And Project 3 in Cardiff is mainstreaming genome-based investigations across specialties, and that is to research the ethical, communication and counselling aspects of incorporating genome-based investigations into the practice of mainstream specialties. So how are we all doing? I'll update you on what we've been up to, what progress we've made and what's coming next. So firstly then, from the team in Manchester on impact and outcome in rare condition clinical trials. So our key updates are really after some frustrating delays waiting for various approvals, we are now conducting our qualitative interviews. So that's really exciting. So we are currently conducting qualitative interviews with parents and carers of children who are enrolled on clinical trials for rare genetic metabolic conditions. And so we've been interviewing those parents and carers about their experiences of having their child involved in a clinical trial. These are kind of open-ended qualitative interviews with the aim of hearing about their experiences and trying to translate some of that data into knowledge about what really matters to patients and children on these clinical trials. We have also completed some systematic reviews as part of our team, and those systematic reviews were on patient or parent experiences of gene therapy clinical trials. Any of you who were at the BSGM annual meeting on Wednesday this week might have seen our poster on that. And that was a really good piece of work that kind of allowed us to further focus what we're hoping to look at with our primary qualitative data collection too. Some of the kind of take-home messages from those systematic reviews was that there is a significant psychosocial impact of participating in a gene therapy clinical trial. And there's a bit of a paucity of qualitative research on this so far, so it's really important that we explore that further. We also found that parents of children who were enrolled in clinical trials have really important perspectives and insights, and actually using those parents could better inform the development of more robust and patient-centred clinical trials as well. We are also currently liaising with more clinicians and more investigators and hoping to set up qualitative interviews with the patients or parents on their clinical trials too. And that's for a wide range of rare condition clinical trials. So hoping to partner with those investigators and do some qualitative interviews with those patients and participants as well. Our pipeline plans then, after the poster presentation at the conference this week, we are also hoping to submit those systematic reviews for publication in journals. And we are also currently in the process of submitting amendments to our ethics approval, which will enable us to expand our study. We have realised that there's a need to actually explore professionals' experiences of working in clinical trials as well, and some sort of barriers and facilitators that they might be able to help us with. We're also planning to conduct a systematic review of this, really just to guide, again, the primary work that we're hoping to do. And we do have a protocol for that on the Prospero website for our systematic review plans here. We also, based on conversations with colleagues, have come to thinking that it's going to be important to understand the experiences of individuals who, for whatever reason, do not take part in or withdraw from clinical trials, as they're also a really great fountain of knowledge about how we could improve that process and perhaps make it more accessible for people. In terms of the PPIE activities that we've been doing, we hosted the first LC conference in March of this year, and I'll tell you a bit about that at the end of the presentation. That has been summarised in an online post that you can get on LinkedIn. And we are, with regards to the sort of study of clinical trial professionals that we're hoping to do, we have been gathering some informal input from professionals who are involved in clinical trials in terms of what they feel it might be important for us to explore or what they're curious about when it comes to this area of research and practice. So that's us in Manchester. Project 2 over in Oxford, their key updates are that their project was launched on the 1st of September 2025. They are currently applying for ethical approval, and that's been submitted and is under review. And they are currently, or they have completed actually, sourcing and collating datasets for a qualitative secondary analysis. So their pipeline plans are to do a large-scale or big qual secondary analysis of around 150 interview transcripts, holding a workshop with key individuals and support organisations, a new piece of empirical research comprising of 20 individuals and families that are living in limbo, so those people who are living without a diagnosis currently for their rare condition. And they're also planning to do a synthesis of qualitative secondary analysis and new empirical work. The activities that they have been involved in are a Making Progress with Newborn screening event, and that was in February of this year, and an event at the end of last year called Songs of Genomics, which is a musical exploration of genomics in healthcare in collaboration with a choir. So some really great PPIE work there. In Cardiff then, the mainstreaming genomics across specialties. Their key updates are a completed ethics application with 4 recruitment sites, collection of 160 lab reports, completing 46 interviews and their 42 professionals and 4 support groups, and presenting a research paper at the Medical Sociology Conference. And what is happening next? They're conducting a validation workshop soon, and that will explore interpretations and analysis. Planning to close recruitment in January next year. Conducting a small number of focus groups with clinicians in key clinical professions over the winter of 2025 and into 2026, and hoping to complete data collection by May next year. PPIE-wise, they've presented at the Wales Genomic Cafe, and they've held a workshop in collaboration with the Brochure Foundation on rare diseases, genomics and justice. So a bit of an overview of our activities and our achievements up until now. So I don't expect you to kind of read all of this just now, but here's an overview of what we've been up to. So the first column there is our PPIE activities, and again, there's the conference there, which really was kind of realising our vision of the LC Node and what we hope to achieve with it. So that was a really good day, and I'll tell you a bit more about that later. We've got an ethics in genetic counselling course for the Cardiff MSc. We had a presence at ESHG 2025 with the genetics workshop. We, as I said, we had some more PPIE activities over the year, which have been really varied and interesting. And then we've also, between us, had presentations as well. So that's been really positive. So just to tell you a bit more about the conference, this was the day after the main RDR UK conference, which was in March in Manchester this year. So the LC conference, we wanted it to be really patient-centred, and it was. So we were very proud that there was a strong cohort of patients and family members in attendance as well. It was really patient-centred, and we wanted it to be really interactive and a day of discussion. So we had panel discussions and lots of audience participation as well. We had a plenary session on paediatric rare condition clinical trials and what matters to children, young people and families. So again, feeding more into our work on clinical trials there and making those more patient-centred. And we also had a panel discussion about patients and families' priorities about clinical trials and treatments. So we went away and did a piece of kind of analysis, I suppose, on the main themes that came out of that conference. And these were equality, equity, diversity and inclusion and ethical conditions in clinical trials, issues around children and communication and consent there, patient and public involvement, the importance of trust and the relationship between patients and healthcare professionals, decision-making and motivations for taking part in clinical trials, experiences of taking part in clinical trials, the importance of clear, accessible and open communication, and patient-reported outcome measures, or PROMs. And I think this really sums up kind of the take-home message from the day, which is that we know about the diagnostic odyssey for rare diseases. And with an odyssey, you don't know where you're going to end up, and it's lots of twists and turns along the way. What we don't want is for clinical trials and research to be another odyssey for families with rare conditions. And if we can make it more of a journey where there's a pathway, where there's support, and where you have some idea of where you're going, then I think that will serve families better. And that was a lovely quote from one of our panelists on the day. If you are interested in reading more about this in full, there is the post that I mentioned, which was actually a collaborative piece of work with us and our kind of patients and families who participated in the day as well. So quite a nice overview there of the day and kind of what we're hoping to achieve as a node as well. So I think that brings me to the end of my presentation. So thank you very much for listening, and I'm looking forward to hearing from the rest of you as well.
Brilliant. Thank you so much, and kind of welcome to the family with all of this.
Thank you.
I just think it's brilliant, actually. You know, the first 2 presentations just exemplifies the diversity of what we do in all of this, which I think is really interesting. I think that's a real strength. And the next is Histio Node, which I believe is Matt Collins, my colleague in Newcastle, but I might have been fed duff information.
It's me. Can you hear me okay? I've got various microphones. Does that work?
Yes, we can.
Okay, great. Thanks. Thanks, Nikita. I think you're controlling the slides, aren't you?
Yeah, Mac. Just
Great.
let me know when you want to shift slides and I will
next
come
one
in.
then. Next one.
Okay.
Great. So I'm going to just give a brief introduction, a reminder of what we're about, and a couple of highlights, a bit of mention on PPIE as well, and a few other thoughts at the end. Thanks. Next one. So we're two groups of diseases that are linked by macrophage madness. The first one is hemophagocytic lymphohistiocytosis, or HLH, which essentially is a TMI, too much inflammation, causes a high-risk disease characterized by cytopenia and a kind of sepsis-like syndrome. So it has a high mortality, it involves lots of hospital disciplines, and the goals are really to understand the triggers better so that we can diagnose it earlier and manage it more effectively. And then we have histiocytic neoplasms, which are a collection of rare inflammatory myeloid neoplasms, so a rare type of cancer. They involve dendritic cells and macrophages, so they're basically cousins of the myeloproliferative neoplasms like myelofibrosis and essential thrombocythemia. And the goals there are to map the genetics and to understand the evolution in more detail, and actually a lot of activity trying to improve access to drugs, because there are very effective drugs because these are molecularly characterized, but the drugs are not currently available on the NHS. Next slide, please. So I need to increase the exposure of the team. Obviously, I had some pictures that I added too late for this one, so I'll skip over. Next one. Great. So I'm just going to give you 4 quick highlights. Fiona Pearce has had a paper accepted at Lancet Rheumatology describing the incidence in Europe. I'll show you a few pictures from that. There's a lot of activity at the UCL HLH Centre with Liz Jury and Jessica Manson, and a couple of students that have been recruited talking about metabolic dysregulation and multi-omic analysis of HLH. We'll show you a bit of data on that. We're just about to submit a big study with European colleagues on molecular monitoring in pediatric LCH. And we've got some new data on the clonal origin of multisystem histiocytosis in adults, which was recently presented at a meeting this year. Next slide, please. So this is the paper that was recently accepted by Lancet Rheumatology. Next, please. And the interesting thing is it shows that there's an apparent increase in the incidence of HLH across Europe over the last decade, and this is primarily driven by greater awareness and more efficient diagnosis. So we're not quite sure we've reached the plateau in this, but this is really a major barrier to improving care in HLH, is to actually recognize the onset of the disease. Next slide, please. And you can see it has, traditionally HLH is categorized as primary and secondary, and you see quite nicely the peak in early childhood, which reflects the incidence of primary HLH driven by inborn errors of immunity, and then this rather large and previously slightly undertreated and neglected peak of secondary HLH in adults, which has a number of different triggers. So a significant burden of disease in older patients. Next slide, please. And this interesting plot of the sort of density of cases across Europe really reflects this ascertainment bias that you saw with the progression over the last decade. So whether you get diagnosed with HLH is really a key factor in whether you get effective treatment. Next slide. Great. So we need to continue to work to increase the awareness of this condition so that we can implement evidence-based guidelines. And the UK team led by Rachel Tafsell in Sheffield and Jessica Manson in UCL are really kind of global leaders in providing clinical, multidisciplinary working and effective guideline management of patients. They actually have their NDT has people from all over the world dialing into them. So it's a really a flagship effort there. Next slide, please. Great. So the UCL team have also been working hard on the sort of cutting edge science, and they've applied metabolomics and transcriptomics to trying to dissect the different groups of HLH, because there are different triggers in adults. So to look at the differences and also the commonalities with ideas of improving biomarkers, and also looking for new pathways to treat these conditions. Next slide, please. So this is I think a Venn diagram of gene expression in the blood, and you can see the sort of these HLH varieties are the same in the middle, that there's a lot of overlap with some pathways, but they also have their own sort of trigger specific differences. Next slide, please. And what they've been very successful in doing is developing a multi-omic signature, which is shown in the top half of the circle, which links to a number of established biomarkers such as ferritin and triglycerides and low blood counts, platelet, lymphocyte, hemoglobin indicated in the blue bands. And this set of sort of gene and metabolic variables is proving very interesting for further study. Next slide, please. So you can see one of these collections of factors is able to associates very strongly with patients that were hospitalized and poorly versus those that have recovered and gone home. So potentially holds some prognostic indicators. It's a very complicated data set, but in amongst this, there are some metabolic pathways which have targetable pathways and genes and drugs that could be applied to actually treating these patients. So really, really exciting research that has potentially a long way to go still, and lots of things to explore. So that's what's happening in HLH. And then we'll just switch to a couple of things in the neoplastic field. Next slide, please. So this shows our study relating to the origin of these rare clonal histocytic neoplasms in adults. We do a thing which you can do with blood cancers where you clone up stem cells from the blood, and if some of the stem cells contain the mutations driving the neoplasia, you can actually go backwards in time and ask in an individual patient, when did they first get the disease, which can be many, many years before the clinical presentation. Next slide, please. And this just shows one of the examples. So this was a patient who sadly died in their late 50s of combined Langerhans histiocytosis and Erdheim-Chester disease. When we look back in the clonal architecture of their neoplasm, we can see that they were living with a triple MAP kinase pathway activation in the blue, green and red bars for about 20 years before they died. And before that, in their 20s, they developed a big clonal hematopoiesis with biallelic TET2. So one of the main, there's lots of interesting features about this, but one of the things that's cool for us is that it anchors the histiocytic neoplasms firmly in the territory of more common blood cancers. So kind of bringing the disease into the fold. Next slide, please. And then finally, some data at the other end of the age spectrum. These are young children with Langerhans cell histiocytosis, and we're looking at the causative mutations in cell-free DNA in the blood, and the pie charts essentially show the frequency of mutation detection in the blood and how it associates with different stages of disease. So moving left to right, the children are having more severe manifestations of the disease. And in the most severe cases, the pie chart on the right, you can see that we can detect the mutation in almost all children, which is going to be a fantastic diagnostic aid. And the level of mutation also increases with the severity of the presentation, as shown on the right-hand side. Next slide, please. And this information, the level of mutation in the blood at diagnosis in the child before they've even had even a radiograph or anything, can give quite powerful prognostic information about their event-free survival. So there's a pivot point in the treatment of histiocytosis because of targeted MAP kinase inhibitors, and future trials will really depend on this kind of information in extracting the higher risk patients and targeting them with a more modern set of therapeutics. So we think this is going to be very impactful. Next slide, please. So we've had a couple of 6 monthly meetings to review our progress. Our PPIE sort of partner, Histio UK, which is a patient advocacy organisation, where it's come to the end of its first generation of founders, and we're essentially supporting its reconfiguration to kind of bring it into the 21st century in terms of all kinds of things, supporting the database activities, reconfiguring its website, even helping its kind of fundraising activities. So that's really a work in progress, but a really critical time for this charity. We've also been involved individually and collectively in pressure groups to get access for inhibitors for histiocytic neoplasms through NHS England, and actually MPs' questions in Parliament, etc. Next slide, please. And the final slide. We really had a bit of a setback in that we were hoping to use a research tissue bank to drive our research, but we just cannot get this on the portfolio and can't get it adopted. So we've had to put our whole project back through REC as a kind of self-contained study. And as you'll be aware, there's a bit of a funding crisis in the NHS affecting all the routes for access to drugs. We spent a long time waiting for specialised commissioning before it was finally frozen. There's no IFR option. We can't get to NICE. But on the hopeful side, we had a recent meeting last week with a LifeArc team in Birmingham CTRU and a drug company to discuss the trial design. There is a bill going through the Lords right now on rare cancer treatments, and I need to read it. I only was made aware of that yesterday. And as Dave mentioned previously, the MHRA are relaxing their market authorization requirements, but yet to be determined how that impacts on our disease. I suspect that's more of a kind of medium-term, long-term thing that will make a difference to the field. And last slide. Thanks to everybody connected with the Rare Disease platform, and also a number of other funders we've managed to lean on to also support this work. And thanks. I'll be happy to take a couple of questions. Hopefully I've left enough time.
You have. Thank you very much, Matt. Okay. Well, thank you very much. We'll move on to the mTOR node. Joe.
Hiya. Can you hear me okay? Am I coming through?
Yeah.
Yeah, brilliant. Okay. Thanks, Dave. So, yes, I'm going to tell you about our nodes work over the last year. So if we can go to the next slide, please. So our nodes focus around a signaling pathway called the mTOR pathway. That's an intracellular molecular pathway that regulates a number of different really key cellular processes. You can see here things like autophagy, glucose metabolism, cell growth and proliferation. So it's normally important, its basic functions to maintain normal cell physiology. And it centers around this kinase called mTOR, which you can see. I don't know if you can see can I get a pointer on here? Oh, no, you're controlling on you, so I can't. So if you look to the left, there's mTOR. So there's a lot of different proteins feeding into this pathway. And obviously the reason that we're working on it here is because mutations in a number of these cause rare diseases. Can you click through to the next panel? So there's about 14 different rare and ultra rare diseases in this group. By far the most common is a disease called TSC, or tuberous sclerosis complex. And one feature of these diseases is they're complex and they affect multiple systems and organs, so it makes it particularly difficult to treat them. The different clinical specialities have to be involved, and it makes it really challenging for patients. So we built a network of researchers and clinicians and patient organisations through the work of the node that are all interested in these diseases and all either trying to understand the causes or develop new treatments or treat patients and help patients in some way. So that's the kind of group of our node. Can you have the next slide, please? Our enabling science projects are, the first one's under the banner Pathways to Treatment, and this is really developing a patient registry for mTOR pathway diseases, which doesn't currently exist. So we're trying to make progress on that, and this is working with an organisation called the National Disease Registration Service. Next point, please. And the second aim is about mechanism. So this is much more the basic science, which I'll show you some data on. We've been doing phosphoproteomics from patient tissue from some of the mTOR pathway diseases patients. And then the second arm of that is to develop a panel of blood cells of PBMCs from mTOR pathway disease patients, and to use the PBMCs from TSC patients to generate iPSCs and develop cerebral organoid models. And the third aim is about improving diagnosis. I won't go into this today. This is something that's taken longer to get going through the node, and ethics has taken a while. So I'll probably leave that to next time to tell you about. Next slide, please. So the patient registry is a big part of the work that we're doing. As I said, we have we're doing this through, rather than a traditional patient registry where we recruit patients together with clinicians and collect information, we're going about this in a different way, working with the NDRS. And we have Layla, who's a research assistant, who has an honorary contract with the NDRS. So she's able to access all NHS patient records. So the progress on that so far, working with for TSC, which is the largest, most common of the rare diseases in the group and has the largest number of patients. We've been working with the clinical partners, about 14 different clinics around the UK, and recruiting patients into NDRS. And we've got over 1,000 patients that we have patient IDs and information from. And that's all now included in the new NDRS rare disease data set. The other thing that we can do with NDRS is interrogate the different databases they have, and one of the main ones is the hospital episode statistics, HES data. And this has worked really well for TSC, so we're able to use the ICD-10 code for TSC and identify nearly 4,000 TSC patients through NDRS, which is by far the biggest cohort of TSC patients that's, I think, ever been worked with. So we're really excited about that, and we just started to look at the demographics, which you can see here, the ethnicity of these 4,000 patients. And the next thing, please. So this is just the TSC patients broken down by sex and age, so we can see the distribution. And we've got lots of different things that we're working on now with this, is looking into things like the drugs that the patient's been treated with, and comparing these data to overall ONS population data to see how they compare, and looking at comorbidities. So we're hoping to get this together for a publication in the next few months. Next slide, please. The next enabling science aim is some of the basic science is doing phosphoproteomics. So the reason for this is that mTOR is a kinase. It's a protein that phosphorylates target proteins, it adds phosphate groups. And so the way to study that at the cellular level is to do phosphoproteomics to, in an unbiased way, identify all of the targets of mTOR. And this has been done in cell culture models before, but hasn't really been done in patient tissue. So we've been accessing patient tissue from various repositories and putting it through our phosphoproteomics pipeline, which we do with a collaborator at the Francis Crick Institute. And this involves doing phosphoproteomics and proteomics, doing a lot of mass spec, and this has been really exciting. Some challenges, because patient tissue can be a lot more heterogeneous compared to cell and animal models, but it's been really interesting project, and I think we're making real progress on this. So the next panel. And this is a volcano plot to show you some of the data that we've generated so far. All the blue dots are phosphopeptides that are significantly increased or decreased in, this is actually TSC patient brain tissue. And there's lots of really interesting things going on here, lots of changes in interesting types of proteins, things that we expected, but also things that we really didn't expect and are very new. And so this is giving us a lot of insight and something we're hoping to get to completion quite soon for one of the projects. Yeah, go on to the next one, thanks. The other part of this is collecting blood from patients. So we had ethical approval in January, and so far out of the 16 patients that we're targeting to collect, we've got blood from 10, might be a couple more after today, and we've isolated PBMCs, blood cells from those patients, and with a company in Oxford StemTech, who are doing the reprogramming part of this. They've taken the blood cells and reprogrammed those into iPSCs, and you can see a nice picture of some of those lines here. And then in January, the Oxford StemTech, and together with us at King's, will start developing, differentiating the iPSCs and growing these mini brains, the cortical organoids, and we're going to be studying the activity of those and the changes of those in the patients, in different patient groups, which we've collected the blood from. And that's just a pretty picture of a cerebral organoid, which we're hoping to produce in the next couple of months. Next slide. The PPIE work has been going really well. It's led by Charlotte Tye at King's. So we've recruited an advisory panel of people with lived experience, which we call the MAP. And the MAP have been really engaged and been helping us with various activities with the ethics applications, which they've given us advice on. And we got them involved in a review article that we wrote and helped them write. They helped us write a section on PPIE, and we included them as co-authors on the article, which was really nice to do. We had a hybrid PPIE workshop in November last year, which went really well. Lots of discussion, lots of sticky notes, which we're particularly keen on these charts you can see there. I think, is there another image? Yeah, oh yeah, so this is really nice, was just unsolicited comments from several of the MAP members when they were writing the review about their thoughts working with the node. You can see that I feel personally this project doesn't just empower, makes you feel you're doing something positive when where you can feel alone and incapacitated. Such complex medical conditions can leave you feeling unable to affect positive change, whereas being involved in a project such as this one gives you purpose. I thought that was a really nice reflection on the benefits that can come to people with lived experience in working with the node and the platform overall. The events, we've been quite busy. We've been giving presentations at patient organisation open days for TSC and LAM. We've given lectures at different organisations at St George's. Sid invited me to Manchester to give a talk there. And there was a mTOR conference in Nice that I spoke at a couple of weeks ago and flagged up the work that we're doing through the node. That was quite a fun picture there. Is there another point, or is that it? Is it the next? Oh yeah, and our next node meeting, we had a couple already. We had a big conference in April 2024, and we've had a bit of a kind of gap to get on with the other things we're doing, but we've got the next meeting conference we're planning for early summer next year to update everyone on the progress that we're making. And publications, we got everyone involved in writing this review article to really define this group of 14 diseases, which hasn't been done before in the literature, and talk about the whole range of activities and the challenges that we're working on in the node. So that was very nice to get everybody in the literature together on the same paper. And as I mentioned, some of the members of the MAP, or I think there's 5 authors, are MAP members on the review article. And there's the QR code if you're interested in taking a look at that. I think that's all I've got to say. So thanks for listening and happy to take questions.
We will move on to the last node, which is Captivate.
Cool. Can everyone hear me?
Yes.
Can I advance slides myself? Yeah, okay. Cool. So, hi everyone. I'm Alex Wimbush. I'm a research fellow on the Captivate node at Birmingham. Just as a very quick kind of overview of the team, there's sort of like the core team, Cindy, Stephen, Tori, myself, Matt, Tom, and Zoe. But we have involvement from a lot of different places for reasons I'll get into. So what is Captivate and what do we see as the utility of the node? There's sort of 2 things I break it down into. The first is we want to sort of be a go-between between people who would want to be running rare disease trials but are frustrated at some of the limitations of conventional trial designs, and they don't necessarily have access to the tools or the expertise to know what alternatives are available and how you go about safely implementing these novel trial designs. And to that end, we want to kind of bring in experience from researchers, from trialists, from patients, and develop a toolbox to make more innovative trial designs accessible. As part of that, there's also 3 sort of research projects we're doing specifically looking at novel trial designs. We want to be looking at how we can develop one-stop study designs, transitioning seamlessly between phases. We want to see what we can do with external information, like what are the limits of how we can safely use external information. And finally, sample sizes. How low can we actually go getting into things like N of 1 studies and how we use N of 1 studies? So just a couple of achievements so far. We got funding and completed a pre-doctoral fellow covering N of 1 studies and different aspects of how people might do pooling in N of 1 studies. We've been invited to speak at many different events, such as the International Drug Repurposing Conference, the annual Wolfram Symposium, the 4th annual CRIC Rare Disease Conference, and we delivered a webinar with the International Network of Agencies for Health Technologies Assessment on the rare disease kind of landscape as a whole in terms of trial design. And we delivered a seminar on calculating with some novel confidence structures and different approaches to statistics at the MRC BSU. So as a sort of like a general research update on what we're doing at the moment and where things are at, a notion called totality of evidence is a bit of a focus. And this is looking at, in rare diseases, if we can collect a range of different measures, patient-reported measures and other measures, is there a safe and meaningful way to be combining this information into a combined outcome that would give us more information, more qualitative information on the impact of a treatment versus just collecting primary outcomes? And there's 2 projects that are kind of ongoing. There's one with Queen Mary University London, and that has been shortlisted for MRC funding. And there's one with York that we're trying to get off the ground at the moment as well. We're also looking at, on the topic of trying to use more qualitative data, there's a doctoral fellowship that has been started by Zoe, looking at how we can bring qualitative data in, because rare diseases offer sort of an opportunity to use more rich qualitative data sets. How we can bring that in whilst addressing the optimism bias that might be inherent in data sets like that in a statistically safe way. And I'm focused on confidence and confidence procedures, sort of as a way of trying to develop statistical procedures that are like Bayesian updating, but have more of the safe kind of frequentist characteristics that we might want out of an inferential procedure. And looking at what that enables us to do in terms of sequential tests as well that have safe operating characteristics. PPI activities, we ran a PPIE workshop specifically looking at patient engagement in trial designs, and this covered 3 sessions. We were initially looking at outcome measures and how people felt about outcome measures. We were discussing perceptions of uncertainty and what it meant for trials to be delivered with maybe different power and error rates. And finally, we were looking at specifically trial designs and how people felt about things like more complex designs, like crossover designs or step wedge designs. And it was a small group, but there was a lot of really, really useful information that came out of it. And I think like the middle quote is one of the things that really stuck out to me in terms of people's perceptions of randomization. And there's this thought that, things like 2-to-1 randomization is preferred because you have a lower chance of being put on the placebo, but it's not that there is a one-size-fits-all approach. In this particular group, there were a lot of people who felt like, well, actually, 50-50, it felt fair. It felt like this compared to this. And I just thought that was very interesting as a very different perspective on the way people think about randomization. So the PPIE workshop was the first of what we were planning to be a series of 3 workshops, and each one we were kind of bundling the information together into a publication. So the PPIE workshop was held with patients and carers, and we've also held a workshop with trialists and methodologists to kind of get a lay of the land and the perception of, okay, what are some of the core requirements and some of the blockers in clinical trial designs in rare diseases at the moment that need addressing, what contemporary research has been done to try and address this, and where are the gaps. and that is being worked into a second paper covering what we found in that workshop. The third is we're on the organising committee for an upcoming UK Regulatory and Access Frameworks and Rare Diseases workshop. And we'll be delivering a breakout session there on trial design and conduct for robust decision-making in rare diseases. We've also been delivering training sessions with LifeArc on Bayesian methods in clinical trials and how Bayesian methods can be used in a variety of innovative clinical trial designs in the rare disease setting. So publications-wise, we've got the PPIE workshop was sent off to Orphanet in July. It's still with Orphanet. We're having a bit of trouble getting that over the line. The second paper from the workshop is following a similar format. We're just in the process of putting that together at the moment. And then I have a few papers in first draft stage on various different forms of conference structures and how these apply to clinical trials, also medical studies generally. And my colleague, Matt Bursnell, also has a paper specifically looking at how we begin to approach totality of evidence in the context of clinical trials and what it means to statistically assess outcomes like that. We had a grant application submitted for a trial designed by Captivate, working with ARDT, that actually made use of the totality of evidence, and that was submitted to the EU Horizon 2025. We have another application that was submitted for a repurposed step wedge trial designed with Oxford, and we're working with LifeArc ARDT, the Rare Therapies Launchpad, to try and enable some novel approaches to clinical design with combinations of N of 1 trials, perhaps. And yeah, that's my overview of Captivate. Does anyone have any questions?
Okay, brilliant, thank you. No more comments. Let's close it and give everybody 10 minutes back. When is the next one of these, Anna? The second one is...
Yeah, Nikki just put it in the chat for us there. So it's on the 25th of November at 1 p.m. and we have our 5 other nodes joining us then.
Brilliant. Okay, so everyone have a nice weekend and thank you for participating. So thank you all.